Another consequence of a damaged glycocalyx is shown here in this slide, and this is on the endothelial nitric oxide production. So on the vertical axis, we measured the amount of nitric oxide produced by the vessel wall endothelium, and then again, we increased the blood flow flowing through isolated arteries. And we could control low blood flows and high blood flows and measure how much nitric oxide being produced. And if we have a vessel with a thick glycocalyx, being able to sense those flow changes, then if we increase blood flow in a vessel with a thick glycocalyx, there's a lot of increase in nitric oxide production. And then again, if you have a vessel that for some reason has a very thin glycocalyx, potentially after being damaged by diabetes or hypertension or smoking, then if blood flow increases, the endothelium is unable to sense those increases because glycocalyx is gone. And there's hardly any increase in the nitric oxide production by the endothelium in response to blood flow changes. While in contrast, if you expose the same vessels with and without glycocalyx to chemical stimuli like acetylcholine, it shows that upon chemical activation, the endothelium is still able to produce nitric oxide. But the endothelium is still there, it can still produce nitric oxide, but the only thing that changes, if glycocalyx is damaged, the endothelium doesn't respond anymore to the physiological stimulus of increasing blood flow. Well, similarly, other people also did experiments to test the same concept. This is a study done by John Tarbell and colleagues. Here they showed that if blood flows increase over cultured endothelial cells over time, you produce more nitric oxide. If you damage glycocalyx with an enzyme called heparinase, then the level of nitric oxide production is impaired. And again, similarly, as the previous experiment with the chemical stimuli, if they used bradykinin as a chemical stimulus to produce nitric oxide from the endothelium, so the bradykinin-induced nitric oxide release was still the same with and without glycocalyx. So for the bradykinin effect, as for the acetylcholine effect, glycocalyx was not relevant. But for the flow-mediated increase in nitric oxide, you need a glycocalyx. Another experiment nicely illustrating the importance of glycocalyx for endothelial cells to being able to sense blood flow is a study done by Forbes Dewey. Here they looked on top of a culture dish with endothelial cells. So every little dot you see here is one endothelial cell. And when you start culturing endothelial cells in a Petri dish, they are randomly oriented. They look all kinds of different directions. But if you start exposing the endothelial cells to blood flow, so again in the flow chamber from left to right, and blood is flowing over the surface of the endothelium for 24 hours. And after 24 hours, if they are flow sensitive, the endothelial cells all start aligning in the same direction. Well, this is in the presence of a healthy glycocalyx. So if you take off glycocalyx, again with an enzyme called heparinase, then even after 24 hours of blood flow, the endothelial cells do not sense that anything is flowing over their surface. And they have no glycocalyx to be able to sense the shear forces, and the orientation of the cell didn't change.